start with the endoplasmic reticulum, the ER, because most classes got either to this spot or right past it. So if you've already gone through these notes, go ahead and fast forward to the ones where you have blanks and need to listen in to finish, but for the rest of you, let's start here. So the endoplasmic reticulum, the ER, is a network of hollow membrane tubes connecting the nuclear envelope to the cell membrane. Okay, what I'd like to say the most about this is that I compare the ER to a highway. So we're literally having open space or open highways from the nucleus to the outside of the cell. And it's going to be able to transport cell products, proteins, things like that that is made in the ER, and then it's going to ship them down the highway out of the cell. You're going to have two types that we talk about, the rough ER and the smooth ER. And you can see a picture here that the rough ER is the ones with the dots on it, with those ribosomes, that gives it that rough appearance. And the smooth ER does not have them. So if we're going on, the rough ER has ribosomes on its surface, that's giving it the rough appearance. But the beauty about that is ribosomes make proteins. So the rough ER has ribosomes that make proteins, membrane proteins, that are going to be transported out of the cell or to other parts of the cell. Okay, that's the entire purpose. Those ribosomes on the ER, rough ER, makes proteins to be transported through that highway to other parts. The smooth ER, it just lacks the ribosome, so it has that smooth appearance. The thing about it is there's no fun way to say this, otherwise it is what it is. It has three specific jobs that we talk about. It has steroids. It makes membrane lipids, which are steroids. It regulates calcium in muscle cells, and it's going to destroy toxins in the liver. So it's determining which cell type it is in, it performs different jobs. So, but the main thing is it does steroids, regulates calcium, destroys toxins, and you just have to remember that. There's not a lot I can say to make that click with you or anything. Just remember those jobs. Here's a picture of the endomembrane system. It allows you to see how it starts in the nucleus, around the nucleus. Then the highway opens up to the rough ER, rough with ribosomes, making those proteins, and then it moves on into the smooth ER, where it's going to make steroids, regulate calcium, destroy toxins. But it's important to see how it starts around the nucleus, and it makes that highway all the way out to the cell membrane and out of the cell. Now it goes on ribosomes. Remember, they are on the rough ER, and ribosomes are going to make proteins. They themselves are made of proteins and rRNA, and rRNA stands for ribosomal RNA. But we call them the protein factories or the site of protein synthesis, meaning they're going to be joining those amino acids we talked about earlier into those proteins. We learned about that in macromolecules. So ribosomes are where proteins are made. Now ribosomes can be found either on the rough ER, giving it the rough appearance, or they can be freely floating in the cytoplasm. And remember, the cytoplasm is a great medium for chemical reactions, and part of that is actually making proteins. And the ribosome will help make those proteins inside the cytoplasm. The Golgi body, I call this the golden packers. Golden meaning Golgi, and they perform much like a post office. They look like flattened sacks of membrane, or pancakes sometimes, and they modify, sort, and package molecules from the ER, and they're then going to store them or transport them out of the cell. So just like a post office is modifying, sorting, packaging, and then shipping it out. So it's our golden packer. Our Golgi body are the golden packers. They're going to package it and move it out. If you're looking and have your animation or your presentation actually playing, you can see this animation in particular, a protein will be made right in here, and then it'll go through the highway of the ER, and then it'll go through here to the Golgi body, and where it's packaged, sorted, modified, whatever, and then shipped out of the cell. Now it's important to know that the materials are transported from the ER to the Golgi to the cell membrane by vesicles. And a vesicle is just a bubble made out of membrane. And that bubble is able to hold proteins, toxins, ingredients, anything that the cell needs to ship out, get rid of, or needs to be brought in. So a vesicle is simply a bubble made out of membrane. Lysosome. Lysosome is the lysol of the cell. So it contains those digestive enzymes that are going to break down the food and bacteria, worn out cell parts. Pretty much is doing what Lysol does, where it cleans out the cell. So lysosome is the lysol of the cell, where it's going to break down, get rid of, and clean out the cell. Cilia and flagella, this is going to be part of the movement a cell can do. So it's made of proteins, tubes called microtubules again, and it functions in the moving cells, moving fluids around in the cell, moving particles across the cell surface, but it's all about the movement. You got two types we're talking about. Cilia. Cilia are shorter and more numerous on cells like hairs. I like to say that the cilia are like silly little hairs. And then the flagella are longer and fewer, usually 1 to 3, on cells, and they're going to move like a flag in the wind, whipping back and forth. And that flag whipping, that is the flagella. So it's pretty easy to remember these silly little hairs, or the flag in the wind whipping back and forth, the flagella. So just kind of remember those sayings, and that'll help out a lot. Again, with a presentation, you can actually see these in motion. There are other cells, especially amoeba, that's going to use pseudopodia for their movement. And pseudo means fake, and podia is foot. So they are literally using a fake foot for movement. So it's a temporary cytoplasm-filled part of the cell that they throw out there to move around, and then they will surround prey and pull it in and eat it, or they throw that fake foot out there and drag the rest of their body to it. So they throw the fake foot, drag them to it. And so amoebas specifically move through that fake foot or pseudopodia fashion. Vacuoles is the next one. They're fluid-filled sacs for storage. A lot of people say vacuoles are the vacuums of the cell, and that's because they're going to be taking in the water and the nutrients and the waste and the foods, and everything that the cell needs is going to store in that vacuole. Small or even absent in animal cells, not all of them have it, but they play a huge part for the plant cell. And it's called that large central vacuole. And you can see it right here in this picture. It holds a lot of water for the cell, a lot of the nutrients, a large central vacuole. You will not find the vacuole in a bacteria cell because a bacteria is a prokaryote. Pro means no nucleus and no membrane-bound organelles. And by definition, the vacuole is a membrane-bound bubble holding all the stuff. A contractile vacuole. This one's kind of cool. I really do like this one. It's found in unicellular protists like the paramecium. And it's going to regulate water intake by pumping out the excess. That's how it maintains homeostasis. If it was not able to do this, then the cell could swell until it lysed or burst. So the lysing simply means bursting. But think of it this way. You have a turkey baster or a pipette, and you can it has a long tube with a rubber ball on the end of it, and if you squeeze that rubber ball, it's going to shoot fluids out of it. But when you let go of the rubber ball, release it, then it sucks fluids back into it. And so that is exactly how the contractile vacuole will work. It's really cool. If you'll do the PowerPoint through a presentation style, you'll get to see it in action here in this, this slide. Now moving on to plants a little bit more. Chloroplasts are only found in producers because the chloroplast is where photosynthesis happens. They're going to be filled with chlorophyll. That's how I like to say chloroplasts are filled with chlorophyll. And this is where they take the energy from the sun and they make their own food in the form of glucose. You know, they store the energy in those chemical bonds that make up that sugar. So it's really a nice way of just saying they take the sunlight and make their food. Okay. Chloroplasts do contain their own DNA. Remember, we have a nucleus with DNA, we have mitochondria with DNA, and we have chloroplasts with DNA. And that all goes back to evolution, how some bacteria or prokaryote cell joined with another one. They worked out really well, and so they decided to stay and evolve together. But the chloroplast contains those enzymes and pigments. The pigment is the chlorophyll, and that's how they do photosynthesis, by using those pigments in the sunlight. You'll never find it in animal and bacteria cells because we can't make our own food. That's just the way it is. And so that's going to be the last slide. I encourage you to make a Quizlet, a Quiz-Iz, or something to study up and get ready for your quiz. Talk to y'all later.